करके रखे हुए हैं। जी 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 जी, श्योर सर, श्योर सर। Thank you, sir. In the name of the most beneficent and merciful God, may the peace, mercy, and blessing of God be upon you. After that, I want to thank the great man because of whom we have gathered here today to celebrate Darwin's birthday. To celebrate the 291st birthday of Anonymous Darwin, the first Briton who wrote explicitly about evolution, the descent of life from a common ancestor, sexual selection, discussed the analogy of artificial selection as a means of understanding descent with modification and basic concept of what we now refer as homology. It is celebrated with joy and enthusiasm in the field of life science on December 12th. In the word of Shakespeare, I just thank you. And honorable dignities, respective faculty members, dear colleagues, professor, lecturers, researcher, and academicians, it is our honor and pride to warmly welcome you all the sixth annual meeting of the International Association of Geologists and the third International Conference on Research Trend in Life Science and Biotechnology, which are organized by International Association of Geologists and Siddhi Earth Group of Education in collaboration with the Department of Biotechnology, Maharishi Markandeshwar deemed to be University Mulana, Ambala. The theme for the year 2022 is Biological, Physiochemical and Radiological Agent Perspective on Human Health and Disease. I honorably welcome our Honorable Sri Tasrim Kumar Garg, Chancellor, Maharishi Markandeshwar deemed to be University, Mulana, Ambala, Professor Ashok Kumar, Principal MMEC, Mulana, Professor Anil Kumar Sharma, a former postdoctoral fellow and senior research scientist at the University of Inauguration at Chicago and head of the Department of Biotechnology, MMDU, Mulana, Ambala, Haryana. Keynote speakers and chief guest, uh, and extend to welcome our guest of honor and invited speakers. This conference will provide a common platform for distinguished scientists, researchers, and postgraduate and undergraduate students to understand, discuss, and debate to the new development and scientific advancement that will impact future strategy to intervene with various debilitating human diseases, ranging for a mechanistic understanding to prevention, diagnostic, treatment, prognostic, therapy, response monitoring, and eradication. This conference will unfold before the scientific community the latest research trend in reputed of areas related to the disease that plague humanity. It is my hope that the worldwide conference will be able to meet in goal of offering a useful venue for academics, researchers and practitioners to advance knowledge, research and technology for the benefit of humanity. There have always been a divide between those who can effectively utilize technology and those who cannot. Humanity can relate to civil rights and social concern, in other words, individual treating each other with care, compassion, and dignity. while recognizing the other person's common humanity as a result despite what we may have led to believe human rights have no connection to polites or any part of so-called political spectrum. They are the part of our common nature, part of the collective humanity is spoken for by our very existence. They neither belong to any political party or fraction nor to any individual or organization, and thus all are the part of common nature, part of collective humanity spoken for by our very existence. In fact, all academic and researcher and practitioners should think about humanity to create a peaceful society. As a result of humanity, we have better environment and better life. We think that education, research, and technology should be available for all people, including minorities, people with disabilities, and everyone who need assistance. In this realm of knowledge, no matter how much we can do our own, whether it's research or development. It's never enough. As a result, the conference's main goal is to share ideas, and it's believed by that by engaging in the exchange, all parties who may benefit for conference will be able to apply what they learn to their own activities. Finally, my deepest gratitude goes to the advisory board, organizing committee, international scientific committee, institutions, companies, esteemed speakers, delegates, participants, and volunteers visiting from the different educational institutes for sharing their experience and making this conference popular enough to attract scholars from all over the world. This committee has put together an exciting scientific program and is working hard to bring highly respected and internationally known speaker to lead it. Although we try our best to the professional on behalf of the organizing committee, please accept our sincere apologies should there be inconvenience that occur before, during, and after the event. I wish you a very productive conference with exciting and encouraging discussion and exchange of knowledge so that together we can anticipate a future of groundbreaking knowledge, research technology for the humanities. May God bless us all with good health to make this event success and enjoyable. Now I am inviting Ms. Bhavna Paliwal, the co-organizing secretary, to proceed with the program. Please,
Thank you,
Ms.
Siddharth.
Bhavna Paliwal.
Thank you, Siddharth. Am I audible?
Yes, please.
Okay. On the behalf of organizing committee and on my own behalf, I welcome you all to the third International Conference on Recent Trends in Life Science and Biotechnology 2022. Special gratitude to all the guest speakers and guest of honors who gave their precious time to us. This year, the theme for the conference is Biological, Radiological, Physiochemical Agents Perspective on Human Health and Diseases. We choose this topic after considering the current state of the world and how these factors are affecting mankind. We are glad to gather researchers and scientists from different areas to seek their ideas on how to deal with the situation. The emergence of the sustainable world may be prevented by excessive level of hazardous chemicals in the environment. The prevalence of hazards is influenced by a number of factors, including weather patterns, heavy metals in industries, pesticides in agriculture, natural and man-made calamities that severely harm both humans and environment. Natural resources and the environment are under a lot of stress from the contaminations of soil, water, air, noise, plastic, and microplastic. This condition may result in spread of illness and infections like SARS-CoV, which made easier by migration. Exposure to toxic chemicals is probably the most significant type of incidents where the immediate and implementations of medical control will be considered to ensure the proper management of patients. Although the biological and radiological incidents have relative longer symptoms onset time, they also warrant early access to medical expertise and control. And this is where we need to apply biological knowledge. This is the biological systems and organisms we have is not new to humans on relying on microorganisms for thousands of years for making items like yogurt, bread, alcohol, and cheese. New biotic innovations arises every day, and the industries will continue to grow as humans seek to surpass biological challenges with specific research-based solutions. And at the end of the conference, we might get some ideas from you all too. The interest of the international scientific community is clear. I'm pleased to share with you all that last year we were able to reach out delegates from 30 institutes and cover 19 states with 81 participants. And this year, the conference has attracted 200 plus participants from 27 entities. We will hear 64 oral presentations and have the opportunity to see about 27 poster presentations. The subject range from descriptions of new techniques like CBRNs, environmental monitoring, radio ecology, safety of food, isotopic techniques in health and environment, and new developments. I'm sure that selective topics will give you with a wealth of information and opportunities for further discussions. The organizing crew did a great job and will continue to do the same in the future. Concluding, I wish you all, in the name of RTLSB 2022, a successful and enlightening conference. And I would like to call Mr. Sushil Kumar Upadhyay, the convener of the conference, to proceed further. Over to you, sir. Sushil, sir?
Yes, sir. Audible.
Sorry, sir. Sorry to interrupt you. You're not audible.
Yes, sir. Audible, sir.
In my in Microsoft Teams, you are not audible, sir.
Yes, sir. Already, sir. Already share it, sir. Already share. Sir, audible, sir.
Siddharth, not audible. Dr. Sushil is not audible. I think he is muted.
No, sir. Sir is unmuted also.
Yeah,
And
but
I am also visible very well.
no, his camera is okay, but voice is not. It's not audible. Yeah.
Yes, sir. Okay, sir. Sir, actually, last talk by Bhavna from MS team. So it's not an issue for streaming. Yeah, sure, sir. Thank you so much, sir. Now I would like to invite you, Dr. Anil Kumar Sharma, sir, HOD, Department of Biotechnology, MMEC, Mulana.
Hello. Very good morning. Siddharth, I am audible?
Yes, sir, audible.
Okay.
Yes,
A
sir,
very
you're-
good morning to all. Today we are here to commemorate the birthday of Sir Erasmus Darwin. I remember his words, the quote given by him, "The love for all the living creatures is the most noble attribute of a man." So let's pay our tribute to Sir Erasmus Darwin and also his son, Dr. Charles Darwin, who has contributed immensely to the science, especially for the evolution, the kind of work they have done, unparalleled work to the society and devising the theory of evolution. So I, Dr. Anil K. Sharma, on behalf of MMDU Maulana Ambala and my own behalf, profoundly welcome the chief guest for today's inaugural function of this sixth annual meeting of International Association of Zoology and the third international seminar of RTSLB, Recent Trends in Life Sciences and Biotechnology. I profoundly welcome the chief guest for today's inaugural function, Professor Bhupender N. Pandey, who is the Honorable President of Zoological Society of India, and also Professor Ramya Vartharajan, who is the Dean of the Manipur University, Imphal. So I profoundly welcome you, sir, with immense joy and pride. And I also welcome all the esteemed guests of honors, esteemed advisors of the international seminar. My heartfelt thanks to International Association of Zoology, as well as Siddhartha Group of Education and Sharda Institute of Science Technology, Education and Research. My earnest tribute again to Sir Erasmus Darwin as well. You know that as we all are aware that a conference is a gathering of intellectual minds, gathering of people who singly cannot achieve anything, but together they can achieve wonders. So that's what the aim of any conglomeration, conference or seminar is all about, which I understand. And the theme for this year's meeting seminar is very well overlapped with the aims and objectives of any society or organization as we try to understand, discuss and debate over the new developments and scientific advancements that impact the future strategies to intervene with various debilitating human diseases, and in order to find some amicable solutions in the form of either diagnosis, pharmacodiagnosis, prognostics, so as to our society becomes benefited to a larger extent. And ladies and gentlemen, what I see in today's context is that modern biotechnology is really being driven by the advanced tools, and we are into a stage where without tools we cannot employ, we cannot amicably find out solutions which are the need of the hour for our society. So towards that context, I think the theme of our seminar holds significance, and I truly anticipate that these 3 days of learning, learning-centric seminar would be really beneficial for our society, for our faculty, for our delegates, and many useful deliberations will come out of this particular event, which I truly hope. So I pray to the Almighty that we all, including MMDU Maulana, as well as all the collaborating partners, will keep traversing the path of light, right learning curve, and creating a robust work culture. With those words, I wish RTSLB a very happy journey, and truly anticipate that this would be a memorable event for us all. Thank you. Jai Hind, Jai Bharat. Thanks a lot. Over to Siddharth. Siddharth, are you there?
जी sir. Yes.
Okay. Okay.
Thank you so much, sir. May I audible and visible? Yeah. Now, sir,
Yes.
I would like
You
to
are
invite you. Yes, sir.
Siddharth, you are audible. Audible.
Okay, thanks. Now I would like to invite you, Professor Dr. Bhupender N. Pandey, sir. He is former professor and head Department of Geology, Dean Faculty of Science and Coordinator College Development Counseling, Madhab University, Bodh Gaya, and former president of the Selection of Geology, Entomology and Fishery of ISCA for the year 1995 to 1996, and resident of college and post Vezani district Bhagalpur, has a brilliant academic career. Now I would like to invite you, Dr. Professor Bhupender N. Pandey, sir. Please, sir. Yes, sir. Yes, sir. Yes, sir. Very much, very much.
Excuse me, sir, you are not audible.
Siddharth, kindly see he is not audible. Siddharth, kindly see he is not audible.
Thank you so much, sir. Yes, sir. Yes, sir. Uh, Now I would like to invite you, Dr. Professor Anil K. Anal sir, Department of Food Engineering, Asian Institute of Technology from Thailand. Please, sir. Professor Anil Kumar Anal sir. Good morning, sir. Thank you so much, sir. Now I would like to invite you, Professor J.P. Sukla sir, IGNTU Central University, Madhya Pradesh. Please, Professor J.P. Sukla sir. Thank you so much, sir. Thank you so much, sir. Now I would like to invite you, Professor Dilip K. Jha, Director, Curriculum Development Center, Agriculture and Forestry University, Nepal. Please, Professor Dilip K. Jha, sir. Thank you. Thank you so much, sir, and all the chief guest and guest of honor. And now I would like to invite you, Miss Suhasini from Chhatrapati Sahuji Maharaj University, Kanpur, to vote of thanks.
Thank you,
Please,
Siddharth.
Miss
Am I audible?
Suhasini.
Am I audible?
Yes, very much.
Thank you so much, Siddharth. I feel incredibly honored and privileged to have been allowed to extend a vote of thanks on this auspicious occasion. I want to extend my warm regards and heartiest congratulations to our chief patron of the conference, Honorable Chancellor Shri Tarsim Kumar Garg, patron Dr. Vishal Garg, and Dr. L.C. Gupta, and co-patron Professor H.K. Sharma and Professor Rajendra Kumar. I also want to thank our chairperson, Professor Ashok Kumar, Director MMEC Mulana, and Professor J.K. Sharma, Dean Student Welfare, MMDU Mulana.
Miss Suhasini, please remove your headphone. Actually, awaz is sound is very much.
Is it okay now?
Yes.
I would like to thank our chief guest and guest of honor for their kind presence in this program. I deeply thanks to our respected invited delegates, invited speakers, keynote speaker, and plenary speaker. I would like to express gratitude towards the convener, co-convener for their constant guidance. Words are not enough to express the gratitude. I also thank the organizing secretary, co-organizing secretary, technical coordinators, management coordinators for their constant support. I would like to propose a vote of thanks to media, participants, professors, lecturers, participating colleges across all the streams of education from research to science and technology. Last but not the least, I am deeply grateful and appreciate the contribution of Department of Biotechnology, Department of Science and Technology, Government of India, Defense Research and Development Organization, Ministry of Defense, Banasali Vidyapeeth, National Institute of Technology, National Institute of Biologicals, Indian Institute of Technology, Durham University, England, Khalsa College, Patiala, Mumbai University, Manipur University, and many more. I feel proud and thank you all from the bottom of my heart. Now I'd like to invite Engineer Siddharth Dan to introduce our chief guest, Professor Dr. R. Varatranjan. Thank you. Over to you, Siddharth.
Yes, I would like to just a permission from HOD sir. Sir, next session is like start a lecture or some. Thank you so much, sir. Now I would like to invite you, Professor Ramayar Varatranjan, sir. He is Professor and Dean, School of Life Science, Manipur University, Imphal. His education credential, BSc, MSc, PhD from the University of Madurai, Kamraj, Delhi and Madras respectively. Postdoc fellow in the field of insect virology at 1998, sponsored by DBT National Associateship Program at the Great Lakes Forestry Center 1998, and he received DBT National Associate Award at 1995 by DBT New Delhi and Lifetime Membership Award by the Society of Tropical Agriculture New Delhi at 2020. He published 4 books and monographs, that is a catalogue on the insect fauna of Manipur in the Indo-Myanmar Hotspot region published by Manipur Diversity Board, Forestry Headquarter Imphal at 2020, and Butterfly of the Manipur published by Manipur Diversity Board at 2015. He also made a short film on insect migration for the UGC Worldwide countrywide and worldwide classroom by help by EMRC Manipur University. He has supervised 8 research projects sponsored by DST, DVT, MOEF, CSIR and supervised 25 plus PhD students. He has published 75 research papers in UGC recognized peer-reviewed journals, and he is fellow of the Royal Society of Entomology, London. He is the coordinator of the Center of Advancement Study in Life Science, Manipur University from 2019 to 2022, and head of the Department of the Life Science, Society of Plantation Crop, ICAR, National Institute of Ecology, Jawaharlal Nehru University, Association Agricultural Important Insects, Bangalore, and member in the Indian Entomological Society of India, New Delhi, and Association for Plant Protection, IARI, New Delhi. He also serving as a member in the project proposal screening committee of DST, DVT, and MOEF, and served as expert committee member of Manipur and West Bengal Service Commission. He is serving an expert to identify trip insects for different national institutes and universities, beside adjusting PhD thesis across the country and screening research manuscript for both national and international journal of entomology. So now I would like to invite you, Dr. R. Bharatranjan sir. Please, sir.
Good morning. Am I audible, sir, now?
Yes, sir. Yes, sir. Very much audible.
Okay. Honorable Chancellor, Maharishi Markandeswara University, Chairman, MMDU, Principal, MMDU, and the Head of the Department of Biotechnology, Dr. Anil K. Sharma, and also the co-chairperson of the conference, and a host of dignitaries as a guest of honor, like Professor Dilip Jha, Professor Anil Kumar Enel, Dr. Usman, Dr. Deepi Sharma, Professor Shukla, Professor Chaube, Professor Rajendra Kumar, Professor Gosi Patla, and Professor Veena Sharma, and other dignitaries, ladies and gentlemen, faculties, and my beloved students, and other dignitaries. And I'm very grateful to Maharishi Markandeswara University for giving me the opportunity. I really wonder that they are able to do such a big conference covering more than 200 participants virtually. In fact, this is a conglomeration of platform for different purposes. The first purpose is, as somebody mentioned, that it is a tribute to Darwin. And second, we are also celebrating the 6th annual meeting of Indian Association of Zoologists. And third, the third international conference on recent trends in life sciences and biotechnology.
But investing is not used. So when used, Google.
Am I audible, sir?
Not audible.
Okay.
Yes, sir. Audible. Very much audible.
It gives me great pleasure because organizing this kind of a conference is not that easy. When I got the invitation, I was checking up about MMDU and the projection of different academic calendar, academic staff, and their facilities and infrastructure, and all so wonderful. And I am very happy that the MMDU is giving a very, very good platform for academic purposes. And it is a beautiful things that happens when you are close to positivity. So similarly, I like this conference. And this conference is not only for a mere meeting of academic friends, but also for exchange of ideas. And another important motive is a dissemination of knowledge, because whatever we know, we are trying to exchange ourselves with a different spectrum of ideas, which will be useful to faculties and students for carrying out their teaching program in their respective universities. So we are getting a chance of updating our knowledge in the field concerned. And moreover, this seminar, this conference is mainly dealing with human diseases. So a number of participants are waiting for presenting their report, their project results, and so on. They are going to help a lot in diverse fields of medical treatment, and thereby alleviating different type of diseases, so that the human being can live with the least disturbances. Added to that, the recent COVID has given us a lot of input. It has taught a lesson rather, and by which we are trying to find out means and ways of what I call getting a better facility, a better knowledge, a better state of living art, and so on. So this kind of a conference will go a long way in benefiting the human in many ways. And I'm happy that the students and faculty members will surely derive a lot of benefits out of it. I wish this event to achieve a grand success, and this kind of a conference really is the need of the hour. And particularly, I thank MMDU for taking the initiative and organizing this lecture. Thank you. Over to Siddharth. Hello.
Thank you, sir. Thank you so much, sir, for your kind words. And now I would like to invite you next chair session, Dr. Mahesh Kumar Shah, sir, and my PhD supervisor. Dr. Mahesh Kumar Shah, sir, is presently at Dr. B.R. Ambedkar National Institute of Technology, Jalandhar, working as an assistant professor since year 2017. Previously, he worked as an assistant professor at NIT Andhra Pradesh and postdoctoral fellow at IIT Hyderabad. He also worked for a MIET Bhubaneswar as an assistant professor. He is a PhD for one of the most prestigious institutions at NIT Raulkela. During his PhD, he worked on biotechnology and medical engineering development of silk-based biopolymer porous matrix for tissue engineering application. He is recently designed and developed and patent invention entitled single piece full body cover, another patent file in method for extraction of water soluble egg cell membrane protein. Dr. Mahesh Kumar Shah sir has been awarded Outstanding Researcher in Biomaterial and Tissue Engineering for the contribution and achievement in the discipline of engineering by Venus International Foundation, India. It was presented during 6th annual meeting Science and Technology Meet ASTM 2020 on 9th January 2021 at Green Park, Chennai. He has been conferred with I-20R National Eminent Young Researcher Award 2020 by International Institute of Organized Research. The awards are bestowed upon the individual and institute recognized by the organization for their remarkable contribution in education and research around the globe. The organization recognized by Dr. Mahesh Sir for Eminent Young Scientist Award in India for his innovation research on biomaterial and contribution to society during the COVID-19 pandemic. He was invited to deliver an expert talk for TEQIP 3 sponsored workshop on innovation project-based learning in engineering and technology. He awarded winners at Green Thinkers Jet Premier Engineering Award 2019 in 4th International Convention on Interdisciplinary Research for Sustainable Development. He has published two test book for prestigious publications, including while one book in the pipeline. Moreover, his contribution to diverse field for medical biotechnology and industrial pharmaceutical biotechnology is greatly acknowledged. He has more than 50 publications in peer-reviewed journal of national and international repute. He also serving as a member in the Institute Digital Respiratory Committee and serve as a board of the study NITJ. He is serving an Institute Library Advisory Committee, Innovation Club, Unnat Bharat Abhiyan, NBA Accreditations, Department of Purchase Committee, Anti-Ragging Squad. He was also serving as a faculty in charge of Instrumentation, Maintenance and Repair, Cell and Tissue Culture Lab, Molecular Biology Lab, UV Testing Facility. Now I would like to invite you, the next session chair, Dr. Mahesh Kumar Saha sir. Please session this.
Good morning, everyone, and welcome to all the participants, all the guests and chairpersons of other sessions. I'm very much delighted to have the participation in this international conference on recent advancement in life sciences and biotechnology 2022. And thank you, Mr. Siddharth, for a detailed introduction of myself. So further, I would like to introduce Dr. Sunil Babu Kosiptala. So Dr. Sunil Babu Kosiptala is currently an associate professor in the School of Life Sciences, Department of Biotechnology at Baba Saheb Bhimrao Ambedkar University, Lucknow, Uttar Pradesh. So he is the member secretary of the Institutional Ethical Committee, Institutional Biosafety Committee at the same institute, that is the Baba Saheb Bhimrao Ambedkar University, since 2016. Before that, already he has served as a reader and the lecturer in the Department of Biochemistry, Bundelkhand University, Jhansi at UP. His PhD work is associated with the isolation and identification of immunomodulatory compounds from the bark of Avicennia officinalis. So he has been the international visitor of the Japan during the 2010, and he has co-investigated a DBT-funded project costing 24 lakhs and entitled In Silico Search for Potential siRNA Sequence to Develop Gemini Virus Resistance Papaya Virus. And now also he is dealing a DBT-funded project costing 74.88 lakhs and entitled Identification and Functional Analysis of Anti-Apoptotic SCMC miRNAs since 2017, and still the project is going on. He has been awarded the first position in the winter school organized by CMEP in 2008. Also has been the gold medalist for the paper presentation on free papers category in National Conference of Indian Academy of Allergy in 2005, Visakhapatnam, India. It was further sponsored by Talwar Foundation in New Delhi. He is a CSIR UGC NET qualified, AP SLET in biochemistry 1999 and qualified GATE in 1995 with 70.67 percentile, awarded JRF. So he has been so many awards and credits goes to his potentials. So Dr. Babu has also been awarded the merit scholarship by Government of Andhra Pradesh by securing the 6th rank in MSc in biochemistry at Andhra Pradesh in the year 1995. He is also awarded the National Talent Search Examination Scholarship at high school level in JPP High School, Ravi Babu in Andhra Pradesh, and nominated by the Baba Saheb Bhimrao Ambedkar University to attend the first Nobel Science Conclave in India organized by IIIT Allahabad in 2008. He is life member of Indian Immunology Society, life member of the Society of Biological Chemists, life member of the Cytometry Chemist Society India, Association for the Promotion of DNA Fingerprinting and the other DNA technologies, life member of Indian Science Congress, annual member of International Society for Advancement of Cytometry, annual member of European Society of Clinical Microbiology and Infectious Diseases. Presently, his research area is deciphering the role of proto-oncogenes or tumor suppressor genes mutations and their roles in cancer progression in relation to the North Indian population. DNA isolation from human blood and tissues, amplifying the required DNA length. DNA cloning is the routinely work in his lab. He used different bioinformatics tools or algorithm in primer designing and being analyzing allele frequency and SNPs. Besides above, he is parallelly working on the host pathogen interactions at miRNA level, human cytomegalovirus, miRNA's role in viral survival in the human body and their contribution in overcoming the cellular death pathways such as apoptosis, autophagy, and necrosis. So, welcome Sunil Babu sir to this conference, and I hope that with your words and with your networking, all the participants will get benefit from this conference. And so here we welcome Dr. Sunil Babu sir. Please, sir. Over to you, sir.
Sir, are you audible? Thank you so much, Dr. Mahesh Kumar Shah, sir. And I am welcoming you, Dr. Sunil Babu, sir. जी सर, जी sir.
Please let me in. Yeah. Hello, am I audible?
Yes, sir, very much audible.
Thank you very much, sir, Dr. Saha, sir. I thank you, the organizers, especially for giving me the opportunity for results of 10 years. I thank the I feel very to participate in this conference and share my laboratory results. So my screen is visible.
Yes, sir, very much visible.
Visible, sir.
Visible, sir.
Yeah. Okay. Let me extend warm greetings from Babasaheb Bhimrao Ambedkar University, Lucknow, to the organizers, because these organizers have brought scientists from 3 different institutions. We really thank you, sir, for giving the opportunity. So today, for the and all the people, I brought a topic that is human cytomegalovirus microRNA's role on cellular apoptosis. We run this area since 10 years, and we got some leads when we published about this one. First time from the virus which is inhibiting the apoptosis. So let's take the it is a DNA virus. It is having a linear DNA of 230 kilobase pairs. It belongs to the beta herpes. If got infection, it gives the cytomegaly. So the cells are going to grow large in size. That's why this virus has been called as a cytomegaly. And it is going to give the intranuclear inclusions. This is the characteristic feature when we do the histopathology for this virus. So, is going to show different type according to the immune status. Suppose this virus infects individual. The infections, the symptoms are subclinical, and the infection is latent type. If you infect a compromised or immune naive individual, it is reported to cause mononucleosis, HCMV retinitis, pneumonitis, diseases, and recent literature is saying it may also cause the carcinogenesis. So this virus is much going to cause many diseases. So if you see its prevalence, 100% of the people living in the developing country are seropositive to this. Indians being in the developed countries, the human adult population shows 100% seroprevalence. This virus is the virus which has dedicated a significant portion of the DNA in modulating the human. This virus reported to co-evolve with the human host and evades most of the host defense mechanisms. They establish successful latent infections. So, despite the humans having the immune able to cause latent infection, so we are interested in studying how this virus and human interaction. So, how this virus is able to do the latent infections. So, our lab since 10 years we are actions, how this virus is going to infect the individual and survive in the human body. So as bring its own proteins and also the RNA and the microRNA with these three type of molecules. It is the human mechanisms and produce the successful infection. So with these three molecules, it can human antiviral mechanisms such as apoptosis, autophagy, and also supports the immune response. So, on how the HCMV microRNAs are participating and how these HCMV encoded microRNAs evade. So, HCMV microRNAs, according to the MIR-based release, this is the microRNA repository. So, mature microRNAs. Most of these mature microRNAs work was identified by the lab from the Fingray University. After the Piffer et al., these are the two scientists who identified the microRNAs in this virus. And now this virus show 26 mature microRNAs. These microRNAs are dispersed throughout the genome. And if you see, it is going to have the unique long sequence and unique short sequence. In both the regions, the HCMV microRNAs are interspaced. And these microRNAs are really going to play a major role in many cellular pathways. As fine switches, because this virus also bring the proteins. Even the proteins are also going to modulate the human antiviral mechanism. So what the microRNAs again going to do is, this microRNAs is sometimes going to augment the sometimes going to act like a fine switching. So they regulate viral as well as host gene expression. Ultimately, the result is to the virus survival in the body. So early studies how to identify the viral microRNA functions. These were there where the viral microRNA functions were predicted through the comparative bioinformatics. For example, if we are going to see RNA sequence, some of the organ which is going to elegans, it is going and present in the Drosophila, present in the human. So the sequences, if you are going to see, one or two nucleides only are different. So that means if we are going to see the microRNAs, some of the microRNAs they are going to be present from human, fly, and worms in other organisms. If you see the another family from the Red7 family of microRNAs, they are also going to present across the worms. So earlier studies were shown on RNAs which will be present in the other organisms also. So we also initiated our study in identifying the function of the HCMV micro in whether these microRNAs are present in other. But unfortunately, our research shows that are unique. They doesn't exhibit any sequence of homology with any other viral microRNAs as well as the microRNAs of other RNAs. So it became very difficult how to identify the function of the HCV microRNA. So the had identified because whole sequence is biologically not important for the microRNA. The central region in the 5' end of the microRNA is called the seed sequence. If this is a seed sequence, it is going to interact with the microRNA shares the seed sequence similarity. They may probably perform the same functions. So that's why many papers saying that the orthologous nature. So we may not need not to identify the microRNA function. But if the microRNA, if the means identified microRNA is going to share the seed sequence of other known microRNA, means microRNA which function is known. So this is the first paper. This came in the Nature. Had published a paper. In that paper, the HCSM Kaposi sarcoma herpes virus microRNA is going with the human microRNA, and that's why these two microRNAs are going to behave similarly. That's why Kaposi correction was orthologous to the human microRNA. In the similar lines, we also published a paper in 2000. The Epstein-Barr virus microRNA is going to have the same seed sequence with the human microRNA. That has, then we published it the first time that the EBV microRNA is also act like. Then, so what we did is we have taken the bioinformatic approach, that is, we searched whether the human cytomegalovirus microRNA seed sequence is going to share the sequence, because most of the human microRNA functions are validated. But to our surprise, that it the 100% score, but the sequence similarity, if you are going to see in the 5' end, so no microRNA is going to have a. So this made us to think again how we can predict the. So there is no sequence homology. The question is neither whole. The whole sequence may be negative, and even the HMV microRNA doesn't have any homology between any microRNA which function is known. We got in trouble how to decipher the viral microRNA function. So then we searched the papers literature found in 2012 and 2013 papers where HMV microRNA UL112 and HMV inhibits apoptosis. So this has given an idea for us. MicroRNAs they are showing. Well, what is the role of other microRNAs? So apoptosis is an antiviral mechanism. So we started our studying the apoptotic genes. So how to study about this? So we have taken the HCl, we started the total study is divided into in silico and in vitro. So in silico studies, what we have taken is we have taken different apoptotic genes. We collected the 3' UTR regions and HMV microRNAs were probed against is there any binding of apoptotic genes through the microRNA and mRNA binding algorithms. These RNA hybrid and RNA which predicts the microRNA targets. Our result shows. So before going to do that, we all know is going to have the extrinsic pathway, intrinsic pathway, and ER stress induced pathway. So what we have detected the genes from the extrinsic pathway, intrinsic pathway, and ER stress pathway. And for these genes, we have collected, and with the help of the RNA hybrid, we probe any binding site is there for the 3' nuclear regions or not. So there were 138 genes were present in the database, and among these 138 genes, we probed microRNAs, and we collected the 3 best results we are going to show. In these best results, we have gene that is modulator of apoptosis for our further studies. Next, the extrinsic pathway, we collected the IRAK1, and HM in mid-UL73P is potentially binds to the similarly, in the ER stress induced pathway, HM in mid-UL148D is going to have 3' nuclear of ERN1. So these today I am going to discuss about the first one, modulator of apoptosis one functions. So we extended the in vitro studies. In the in vitro studies, what we have done is mimic we procured from the ambient, and we induced the H2O2 in the HAK. Why we have taken hydrogen peroxide in inducing the apoptosis in the cells? Because this hydrogen peroxide this is most similar to the viral infections. Because most of the time, HMV infects the body, the cell is going to undergo the this is the intrinsic pathway of apoptosis. So we outlined the in vitro studies. We have taken the HAK2 the apoptosis through the H2O2 and staurosporine, and we have seen the induction and inhibition of apoptosis through the microscopy. Assay like caspase 3 and 7 assay. In the microscopy, we have seen the apoptotic cell characteristic features like chromatin condensation and membrane building. Now, coming to the H2O2 induced apoptosis, we studied the apoptotic cells and scanning electron microscopy and flow cytometry. And if you are going to see that in the control cells see in the there is here also in the SEM also there is no clear cut membrane webbing. Even in the apoptotic cells also, but the apoptotic cell but when we treated the cells with the H2O2, then we have seen the clear cut nuclear fragmentation. And it shows, it indicates the cells are undergoing the apoptosis. In the scanning electron microscopy, we have seen the membrane cells through the flow cytometry, the apoptotic cell ratio is increased. Now, so we comes in these cells. When we simultaneously treated with our microRNA, which is going to, which we speculated, it is going to inhibit the apoptotic cell ratio is significantly downregulated through the DAPI, even in the microscopy scanning, the membrane webbing. Even in the FACS also, the apoptotic cells, when you compare it with this one, is decreased. Now, next question we ourselves ask, is it due to our microRNA only, that is mid-UL73P only, or any other factor which is responsible for it? We have taken the microRNA inhibitor. So with the simultaneously we use the inhibitor that the apoptosis got reversed. This clearly indicates that the apoptotic inhibition is observed to RNA only. So this established us that mid-UL73P is going to inhibit the H2O2 induced apoptosis. Now, actually, many scientists, when we sent to the journal, they asked biological assay. So then we have done another biological assay where we measured the caspase 3 and 7 levels to the. Here, if you are going to see, suppose we are going to treat the cells with the H2O2, the caspase levels are going to be high. With the microRNA mimic, the caspase levels are downregulated. This clearly shows the HCMV mirrors and may also downregulate the caspase 3 and 7 activity. And we used the inhibitor, then the reversal of the. This confirms that our mirror 73P downregulates the H2O2 induced apoptosis in HEK2. Next, what we have seen is that's okay. But what is the functional target? Our in silico results shows functional target. Now we ask whether this MOP1 mRNA expression is what is the effect if we are going to 73P. What is the expression levels of the MOP1? It clearly shows the MOP downregulated. So we got a confidence that the virtual 73P is going to do by targeting the modulator of apoptosis 1, which happens to be the extensive pathway of apoptosis gene. Now, so to study the binding, whether this microRNA is binding to the 3' UTR, that we made a dual luciferase vector, and in this vector we are going to have the 3' UTR region. So this vector is going to be deleted type. In the deleted type, we remove the MOP1 3' UTR so that for this microRNA bindings are not going to be present. So when we this assay is going to confirm the microRNA binding site on the mRNA. So if this is going to be used, then we have measured the relative luciferase activity through the luminometer, and that is going to be the 100. So when the relative luciferase activity is almost decreased to 60%. So again, whether we are so when we add the inhibitor reverses the luciferase activity. So it clearly demonstrates site in the 3' UTR of MOP1 is functional. Again, we asked ourselves, suppose we have taken UTR which doesn't have any MOP1 MOP1 UTR. So for that also we have taken, and we have in the luciferase activity, showing that the microRNA, if we add also, it doesn't have any effect because into the 3' UTR in this vector, because it is not going to have the any 3' UTR. Even we inhibit also with the so we didn't find any answer. This clearly shows that the microRNA vireo 73P has the functional binding of MOP1. Now, next we will further ask, okay, that is fine. mRNA is there. mRNA is RNA is going to downregulate the protein levels. If you are going to see clearly the HCM vireo 73P, if you are going to there, the MOP1 protein levels are these experiments clearly demonstrates that MOP1 is going to be the functional target for the HCM vireo. We are doing the first time, so the reviewers had asked many questions for us, whether this microRNA mimic is working, whether this microRNA, how much extent it is inhibiting, like that. So we compared the down-regulatory effect of vireo 73P with the SIR and the normal, and we have seen, and both siRNA and microRNA both are going to show the inhibition of and also caspase 3 also they are going to inhibit when compared to the H2O2. And even in the qRT-PCR also, expression is also going to be down-regulated by the both of the things. So we have even gone for the protein expression. This shows both muriel 73P and siRNA of MOP1 down-regulates the MOP1 mRNA levels, apoptosis, and down-regulates the MOP1 protein levels also. But there is a slight thing here, that is as more inhibitory effect than the UL73P. Here also, if you are going to see the siRNA is more inhibitory effect. Here also more inhibitory effect than the microRNA. So the summary of the first study is HCM UL73 to induced apoptosis by translational repression of pro-apoptotic gene MOP1. So again, we have seen the literature. This was down-regulated by the even human microRNAs also. Those are HSC and MIR1228 and HSC. This increases our confidence that even viral microRNA also can inhibit the apoptosis. This is the first work summary. So second case, what we have done is we have seen another microRNA and with the M48D. It is going to inhibit the apoptosis by targeting the another molecule, that is ERN1. ERN1 is nothing but the nucleus signaling one. This molecule is works at the endoplasmic reticulum stress. So we had chosen that is a staurosporine, which actually works at the ER stress. So the same here the apoptotic inducing agent is the staurosporine. So we studied the apoptotic effect and the UL148D through the flow cytometry. Here also we are going to see, if we are going to see the staurosporine with the MIR, the apoptotic cells are going to be decreased. Even in the micro DAPI also, we can see the apoptotic cells. So here also we used the inhibitor of the microRNA inhibitor, and this microRNA that yes, the inhibitory effect observed is due to the microRNA only. So then what we how the apoptosis is going to be inhibited if this microRNA UL148D are if we have the apoptotic genes. What are the apoptotic genes which are going to be enhanced? Here, if you are going to see the ERN many other apoptotic genes are also increased, but the ERN1 also increased. The back one is also increased. But our in silico works predicts that the functional target for the UL148. So then we have seen through the qRT-PCR whether UL148D, what is its effect on the. So when we are going to see the ERN1 mRNA, so the significantly ERN1 mRNA is also decreased. So we are selecting the others because both these apoptotic genes are increased, but down regulation is done by this microRNA. So similarly, we have taken the dual luciferase vector and we studied the binding sites with the mimic luciferase. So here we see significantly seen the microRNA mimic is reducing the luciferase. Of the luciferase activity shows that it is going to inhibit the it is going to bind the 3' Similarly, we have seen the vector which is not having any UTR, so there is no change in the luciferase. So even the protein levels also we have seen. Even these proteins are also we have seen. So RRA on when we are going to put the mimic. So this question came: how this ERN1 molecule stops the apoptosis. How targeting the ERN1 can stop the apoptosis. So we have so ER1 gene is going to encode a IRE1 alpha protein in during your ER state. Alpha protein is going to dimerases. Once it got a dimerases, then it is going to do the unconventional splicing of the XBP. XBP1 unconventional splicing leads to the XBP1 spliced one, and XBP1 spliced product is a transcription factor. Once it goes and binds to the nucleus, it is going to produce the endoplasmic reticulum associated proteins degradation. This what is going to happen is, if any excessive unfolded proteins are there, so IRE1 is going to get activated, and XBP1, and the spliced product of the XBP1 is a transcription factor which is going to release the ERAD molecule, which is going. But the same molecule will perform another function also. If the stress is not resolved, then what happens is the same molecule is going to activate the apoptosis with the help of the TRAF1 and JNK phosphorylations. In this case, we studied on the IRE1. What happens to the XBP1? What happens to be the JNK phosphorylations? So in our next splicing and unsplicing levels of the XBP1 and also the phosphorylations of the JNK. So the results shows the of the XBP1 unspliced and spliced also, if you are going to see. And when we are going to see the mimic, in this mimic, the spliced one levels are decreased. So spliced one is decreased means UL148D spliced portion of the XBP1. So that means it is going to reduce the proteins. So here also, and unspliced XBP1, when you see the unspliced XBP1, when the HCMV meridial 148D is significantly downregulated. This shows us, yes, the downregulating the IRE1 alpha protein. So then we have seen further phosphorylations. What happens to the JNK phosphorylations? Even if you are going to see the JNK phosphorylations, the HCMV meridial 148D levels also. This clearly demonstrates even the phosphorylations of the meridial 148D. So the summary is HCMV meridial 148D downregulates staurosporin in translation repression of ER stress signaling molecule, that is ERN1. Staurosporin induces IRE1 alpha XBP1 splicing. Is downregulated by the our HCMV meridial 148D. So not only this UPR, unfortunately, even some human microRNAs are also reported. They are going to downregulate the clinicamycin-induced UPR by targeting. So this has given a confidence for us. Yes, this microRNA is going to inhibit the that is ER stress-induced apoptosis. Now, regulation apoptosis, if you are going to see. So we have seen many of the microRNAs are reported. This is UL73P and UL148D is our results, but other that the HCMV microRNA is downregulating the apoptosis. But few studies are also reported that some of the microRNA sequence, they are going to upregulate the apoptosis. Now we got it out how we can understand the mechanism. So this gives the microRNA mediated regulation is going to be a complex, and it is going And this complex and due to this complex and spatio-temporal in nature, so apoptotic inhibition when apoptosis, when the virus promotes the apoptosis is different. So according to the literature, it came to know, means it is clear that initial periods of infection is beneficial to the virus. In later stages, it may not. So that was the seen. What happens to the proteins? Because the HCMA brings proteins also. So the literature we have seen, sometimes this HCMA exhibit a synergistic effect. The literature shows, for example, PUL 737X.1, 2.7 RNA. These two RNA as well as the proteins, they are going to disrupt the mitochondrial membrane integrity. To disrupt the mitochondrial integrity proteins. So what will happen is sometimes the apoptosis is inhibited or apoptosis is going to be, we can say, initiated. Our mid-UL 36-5P and UL going to affect the molecules, don't regulate the molecules which are going to show the mitochondrial membrane integrity. So in human cytomegalovirus is going to inhibit the apoptosis through its microRNAs, but how and when apoptosis inhibition or augmentation is required is decided by the virus according to the internal. So I want to acknowledge the team which we are having, the SEM and confocals we have done in the symmetry studies in the CSIR IITR, and there are collaborators in the CSIR IITR, Dr. Sanjay Singh, and is done by the Dr. Abhishek Pandey, my student, who is working and Anup Mishra and Aarti Priyadarshini. I would like to thank the funding agents, because all these works without the funds. So I thank to the DBT Government of India and DST SEB for funding my projects, and of course my parent institution from the to my BBAU Lucknow. Thank you very much for your kind attention.
Thank you, Sunil sir, for all this information that you have observed during your research. And obviously it is the output of the research, but it is the guidance that always matters. And further, I'm just opening the house for some queries from the participants, if any. Any queries from the participants? Okay, so we have not received any response from the participants. So I'm just handing over this to Mr. Siddharth. Thank you, Sunil sir.
Thank you, sir.
Thank you so much, sir.
Thank
It is
you,
very
Siddharth.
fruitful information you can give me and ours. Now I would like to invite Dr. Sushil Kumar Upadhyay, sir. Please proceed the program.
Thank you, Siddharth.
Yes, Thank you, sir. In the other word of Shakespeare, I can't make no other answer than thanks and thanks. And I will again thank you all the guest speakers, chief guest, guest of honors. And now our rule and norms, we can follow the presentation of the invite delegates. First category, our invite delegates, then young scientist oral and young scientist poster. So now the session is 12 o'clock to 1:30 o'clock. That is a proceeding invite delegates lecture. And our first invite delegates is Dr. Jitendra Singhediya sir. I hope Dr. Jitendra Singhediya sir are available in Meet and he is audible. Dr. Jitendra Singhediya sir. Sir, please unmute yourself. Sir, please unmute yourself.
Can I audible, sir?
Yes, yes, yes. Very much audible. Very much audible. Please share your screen.
Yes, sir. Please allow to me share.
Already allowed because you are as a presenter and you can share it.
Sir, where is the option?
After the camera, mic, and just you
I
can
have
check.
share slide button, not screen sharing.
Sir, you can join by 2 account.
Yes, sir. One up on my mobile and second is on computer screen.
Okay, okay, okay.
Can you see my
Yes, yes,
PPT?
yes. Very much, very much, very much.
Yes, sir. First of all, good morning to all of you. Today I am presenting.
Sir, I want to request you, please remove your mobile from the nearby computer or laptop.
Yes, sir. Is this okay?
Yeah, no, no, no.
1 minute, I have left